Atamarie, good morning. I'm Helen Whelan of the Bioprotection Research Centre at Lincoln University, and I'm here today to give an update on the Trichoderma project in New Zealand Forestry. The objective of this project is to develop a long-term symbiotic relationship between the forestry trees and beneficial Trichoderma root endophytes. So we're looking to improve the economic returns by increasing production, and this may lead to shorter rotation times. We're looking to help alleviate the cost of current and future foliar disease epidemics, particularly when we have more impacts of climate change. Trichoderma can also offer a non-chemical product to help reduce the agrochemical use in nurseries and forests. Now, this project is mainly concentrated on radiata pine, although there has been some work done in Douglas fir and cypresses. So I'd just quickly like to review what Trichoderma fungal root endophytes are. They're a naturally occurring fungi found in plant roots and in the soil, and they have a symbiotic and mutualistic relationship with plants, so both are getting benefits. So we've collected hundreds of strains from the roots of very healthy pines, natives, cultivated and weed plants throughout New Zealand, and we've tested them for growth benefits and disease suppression, mainly Dothostroma, terminal crook, and Diplodia in greenhouse and plantation trials. I'd just like to point out that Trichoderma does a similar job to mycorrhiza fungi. They can both improve nutrient uptake, promote plant growth, and reduce the effects of environmental stress. But Trichoderma is particularly good at activating the plant's defense systems, and they can be aggressive against pathogens. So the main difference between Trichoderma and mycorrhiza is that Trichoderma is not visible to the eye, and it's often inside the root, compared to mycorrhiza fungi, which are usually seen outside the root. Now, Trichoderma fungi, they're relatively easy to propagate and they're easy to use in the nursery, with the most cost-effective, practical and efficient application method being as part of a seed coat recipe, although they can be applied as a soil drench with commercial equipment or as a root dip. So there's been good uptake in 2021 with 10 nurseries using inoculum in their production. An important question we have looked at is how tolerant are Trichoderma inoculants to commonly used agrochemicals and other products. So this is information for nurseries that want to continue using agrochemicals. So we did a large study testing. It was 14 fungicides, 2 fumigants, 14 herbicides, 6 insecticides, 4 fertilizers, 5 adjuvants, and 6 adherents, biostimulants, and a bird repellent. So we tested the strains that made up the PR6 and PR3A blends, these being the final 2 blends that were selected throughout the project. Now, these agrochemicals were generally recommended for use in forestry nurseries and plantations, and they were applied at recommended label rates, mainly for nursery use, although a few of the agrochemicals were applied at the higher plantation rates. And if you would like to get the details of this study, it can be found in this report on the FGR website. Now, there were 2 types of experiments done. The first was in the lab, where the agrochemicals were applied to the spores first in solution. So we were looking at whether there was an impact on spore germination. Also, they were mixed in with agar to see if the growth of the fungal mycelium was affected, and we looked at the sporulation as well. Now, these experiments were considered direct contact between the Trichoderma and the agrochemicals, And this gives information on whether spores can be mixed with agrochemicals in spray tanks if you choose to do this. The second set of experiments were 8 greenhouse experiments using either soil or potting mix. The agrochemicals were applied as one application. They were applied at different times, depending on the type of agrochemical. We also did a seed coat experiment. Now, in all cases, the seeds were inoculated with trichoderma at sowing, and obviously the trichoderma grew into the roots over time. Now, we considered these greenhouse experiments to have indirect contact between the trichoderma and the agrochemicals, apart from the seed coat experiment. And over time, we measured the levels of trichoderma in the roots in the presence of these agrochemicals. Now, if you look at the lab experiments with direct contact, we found that the strains were sensitive to 16% of the agrochemicals tested. So we came up with recommendations for spray tank mixing, if you choose to do this. The PR6, PR3A blends, they can be mixed with insecticides, fertilizers, adjuvants, and biostimulants, and most herbicides. And this is because the spore germination and viability was not affected. But we suggest avoid mixing these blends with some fungicides that are listed on the slide, and also boron, and that's because of the potential fungicidal effects on spore germination. Spore germination can be reduced. And the other direct contact experiment was when trichoderma was applied as part of a seed coat recipe. In this study, the trichoderma root levels were measured at 27 days, so 1 month and 2.5 months after the trichoderma and the fungicides were applied to the seeds. And you can see, if you look at the 4 bars on the left-hand side of each graph, that the fungicide treatments had similar or enhanced trichoderma root levels compared to the control, and the controls are the hatch bars on the right. So this was an important positive result because this shows that trichoderma can be safely applied with these fungicides at seed coating. Now, in the indirect contact experiments in the greenhouse, we found that trichoderma was tolerant to all of the agrochemicals tested. So this included the soil prep herbicides that were sprayed before sowing, the soil drench fungicides applied either just before or just after sowing, and the soil drench herbicides at sowing. So this tolerance may have been because the growing media provided a buffer between the trichoderma and the agrochemicals, even when the agrochemicals were moving through the soil. And sometimes we found the trichoderma root levels were enhanced, that was shown in the previous graph, and possibly the trichoderma was able to dominate the other resident fungal species. Trichoderma was also tolerant to aerially applied herbicides, fungicides, and boron sprays, and this may be due to there being a sufficient distance between the trichoderma and the roots and the applied agrochemicals. So overall, this gave us confidence that the selected trichoderma strains are compatible with most of the forestry agrochemicals at standard rates, particularly when they are applied to the growing media. We also did a small amount of work which suggested that these trichodermas were tolerant to agrochemicals at higher rates when they were applied to the growing media. Now, in this section, we are looking at the impact of trichoderma on tree growth and disease suppression in plantations. So we've set up validation trials to test the PR6 and the PR3A blends. These blends were applied to the seed in the nursery, and we are comparing these to the untreated control. There were 8 trials set up in 2018, 2 in Gisborne, Northland, Bay of Plenty and Nelson. They were large scale with a robust design, so there were many trees per plot and a high level of replication. A new series of validation trials has recently been set up in the South Island, looking at whether these Trichoderma blends will perform well in cooler and sometimes drier environments. So they were established in winter 2020 and 2021, with 1 trial in Nelson, 2 trials in Canterbury, and 3 in Otago. Again, they were large scale with a large number of trees per plot and replications. So if we look at the results of the 2018 trials, first in the two graphs on the left-hand side, in year 1 and year 2, the average height was at least 6% higher in the Trichoderma treatments compared to the control. And in the 4 trials where we were able to measure trunk diameter, this is the graph on the right, the diameter was 9 or 10% greater in the Trichoderma treatments. So Trichoderma has given a significant boost to early plantation growth, and we're looking to measure these trials again in year 4, which is next winter. So we're starting to measure foliar disease. It's mainly Dothostroma at this stage in these trials. So this is an example of one of the Kaingaroa trials where there was a 45% reduction in disease severity, and that was measured last November. So this is a promising result so far, but we need more measurements over time. So for the ongoing or future research, we want to continue to quantify the effects of Trichoderma on growth and disease suppression in nurseries and plantation trials. We continue to study whether these strains can be applied to uninoculated forests to increase growth and health, but we need to find a practical way of applying the Trichoderma. Can Trichoderma be used to boost poor or new production soils in nurseries? So we're starting to get good results with this and getting good nursery feedback as well. We need to find out or continue to find out how Trichoderma is persisting in plantation trees, how long it's lasting. And we have projects looking at whether Trichoderma can control canker in cypresses and Swiss needlecast in Douglas fir. We'd like to start to do new research into whether Trichoderma can be used to improve stand uniformity and to show whether these Trichodermas are contributing to increased needle nutrition in the validation trials. So in conclusion, there are early indications that Trichoderma inoculated plantation trees have more growth and less foliar diseases than the untreated trees. And in addition, we've found these Trichoderma blends are beneficial in other forestry species as well. So these Trichoderma blends appear to be a promising, effective growth and biocontrol tool for New Zealand forestry. We found they can be successfully integrated into most production systems that use agrochemicals and other products. And there has been good uptake in nurseries this winter, with inoculum for 60 million seeds being sent out. And this is all part of the process to help evaluate a business case for the commercialisation of a Trichoderma blend for New Zealand forestry. Now, I get a huge amount of help and support from industry partners. So this is a big thank you to the companies that are listed on this slide. So thank you for taking the time to listen to my talk.